We've talked a little bit about ATP so far, and you know that the basic idea is that the molecule adenosine triphosphate, ATP, which sometimes I use the cartoon of the little purple thing with 3 orange dots, those 3 orange dots being the phosphates, can be broken up into ADP plus a separate phosphate. So that's adenosine diphosphate plus the separate phosphate. And it's in that bond between the second and third phosphate that we stored a lot of energy. We've spoken briefly about this, but let's look at this again, but let's look at this in the other direction. Remember, this is a reversible reaction. So it is also possible to make ATP by taking ADP and phosphate. So I can take an adenosine diphosphate and a phosphate and stick them together to form ATP. Now, we've mentioned it before, but I'm going to mention it again. The reason that we get energy out of ATP is because we had to put energy into it to make it. So remember that if I've got my adenosine, my adenine on my ribose with 2 phosphates, those are carrying negative charges, and I bring in another phosphate carrying a negative charge, it doesn't want to go near those other phosphates. It's difficult to get it there because negative charges repel each other. But if I have the right enzyme, I can force those phosphates to get close together. Remember, I have to put energy into it to force them in there, and then make a covalent bond which is going to tie them together. So if I take this and also put in energy, then my adenine, my ribose, phosphate number 1, phosphate number 2, and phosphate number 3. This is the bond that I just formed that ties that on there. If you imagine that bond is almost like a little spring that I've compressed. I had to push that phosphate in and then tie something there to hold them together. But now there's this compressed spring between them. If I cut the thing that's tying them together, bing, it's going to spring off and that phosphate is going to go flying. I'm getting some of the energy back that I put in to get it in there. That's the idea of ATP. What I want to get to here is that you can make ATP again by putting that phosphate on there. We can get energy out of it by breaking the phosphate off. We can make the ATP again by putting energy back in. You always have to put more in than you get out. That's the way things work. So part of what we have to talk about in a muscle cell is how do they make ATP? Muscle cells use a lot of ATP. They use ATP to run their ion pumps. They use it to run the myosin. They're very, very ATP-intensive cells. So where do they get all this ATP? So that's the thing we have to explain here. And there are really several mechanisms we use. Phosphocreatine as a mechanism is going to be useful in the short term. That can be used to generate ATP quickly, but really it's just more like a battery that we stored up that we're then going to use to regenerate our ATP in the short term for seconds to maybe a minute or two. If we need more, then we're going to need to use glucose. Glucose metabolism, taking a molecule of glucose and breaking it up, extracts a lot of energy from the glucose, and we can use that energy to put ADP and phosphates together to make ATP, and then ATP to power the stuff in the cell. But that means we have to take glucose and break it up. So that's the whole process of glucose metabolism. And we can have either anaerobic metabolism or aerobic metabolism. One of them is faster. The other, actually, aerobic has to also do anaerobic. The first steps are anaerobic. Takes longer, more complicated, but generates a lot more ATP. It is also possible to use fatty acids for fuel in muscle cells. Those produce even more ATP. They're actually the most energy efficient fuel, but they take the longest to use. It's a slow process. So we're going to look at all three of these mechanisms and go over how each one works, how much ATP we can get, and what's involved. These are somewhat complicated. So be ready to have lots of room in your notes, and you're probably going to need to write this stuff down and then go back and look at it again, and maybe even a third time. So let's get to it. A muscle cell holds about 8 twitches worth of ATP. So enough ATP to power about 8 twitches. That's not very much. If I start using a muscle to sustain a contraction for any amount of time, I'm going to burn through that 8 twitches really fast. And you know that when we run out of ATP, so if we ever run out, you know that what happens then is rigor mortis. The muscle will lock up. So we never want that to happen. We never want a muscle cell to run completely out of ATP. To avoid that, we're going to need some way of rapidly regenerating ATP when a muscle starts getting used. So the moment it starts burning that ATP, going from ATP to ADP plus phosphate, we're going to need a way to regenerate that ATP really fast right at first. Now, we mentioned several ways of making ATP. We talked about phosphocreatine, glucose metabolism, and fatty acid metabolism. Glucose metabolism happens either using the enzymes in the cell or in the mitochondria if you're doing it aerobically. And it works very well, but it does take a little while to get started. Once you start running glucose through that process, it takes a short time before you really start getting ATP back out of it. When a cell is at rest, it is burning glucose, but relatively slowly. So when you start using a muscle cell, even if it started increasing glucose metabolism quickly at that point, it would take a short time for us to start getting ATP out of that process, which means we need a bridge. We need a way of regenerating ATP quickly while we are waiting for glucose metabolism to get started. And that first method, the short-term method, is phosphocreatine. So phosphocreatine allows rapid regeneration of ATP in the short term. We can use phosphocreatine for seconds to minutes to keep our ATP reserves from dropping to nothing while we're waiting for glucose metabolism to really get started. So let's talk about how that works. So the basic equation for ATP is ATP gets split up into ADP plus phosphate. Now, there's another possibility. So if I have more ATP than I need, say we've got a muscle cell at rest, and it is in the slow rate of glucose metabolism that's going on, it's able to generate more ATP than it needs. So it builds up its 8 twitches of storage. So what's it going to do with any more ATP that it can make? Well, one possibility, let's represent the ATP in the way we were doing, as the purple dot with our 3 orange dots. So there's our ATP. We can bring in a molecule called creatine. And if you bring these together with the right enzyme, we can transfer one of those phosphates to the creatine. So one of our 3 phosphates gets moved to the creatine, and we come away as ADP and phosphocreatine. I'm just going to get rid of this because we don't really need this right now. So this is something we can do when the muscle cell is at rest. And we can end up building a big pile of phosphocreatine. Now that ADP, if we've got other phosphate around, we can take that ATP and run it back through glucose metabolism. Combine it with another phosphate and make some more ATP. This is slow though. So at rest, the only reason we can do this is because we're not using very much ATP relative to what we're making. So if we're making more than we need, we can take some of the excess ATP and use it to transfer a phosphate onto creatine, making phosphocreatine. Now, the reason that that's useful is this. When the muscle is in use and we start we're taking ATP and breaking it up into ADP. So our ATP is being used by the myosin or by calcium pumps or whatever. And we end up with a lot of ADP, so effectively expended ATP. If we take that and combine it with a phosphocreatine, we can transfer that phosphate back from the phosphocreatine to make ATP again. And this just becomes regular creatine again. So think of phosphocreatine as a charged battery pack. So let's say when we're at home and we have relatively good access to power, I charge up my phone on the wall and I charge up a special battery pack. And then while I'm out, when my phone loses its charge, I can take my battery pack, plug it into my phone, and recharge my phone. So my phone is like the ATP. When I'm at home, I can charge it up with glucose metabolism, and I've got a charged phone, and I can also make phosphocreatine. And then when I'm out, I can use the phosphocreatine, the battery pack, to recharge the phone, make ATP again, until I can get back to where I have access to power. The phosphocreatine is just a way of storing power in a non-ATP form that can be used to make ATP quickly. Now, there's an enzyme involved in this. The enzyme that's involved in transferring that phosphate is called creatine kinase. Remember, a kinase is something which phosphorylates something else. So this creatine kinase is adding a phosphate to creatine. So that's what it's doing. Interestingly, the enzyme that does the other direction is also a form of creatine kinase, a closely related form. So it's pretty close to being the same enzyme that can make the reaction go either way. The only advantage to this is that it's very, very fast. You can get ADP and phosphocreatine and transfer that phosphate really quickly. So if you imagine you're a muscle at rest, you've got extra ATP, you make a bunch of phosphocreatine, and then when you're the muscle in use and you've sent your ATP off to the myosin and it comes back as ADP, you can quickly recharge it using phosphocreatine, making it into ATP again. And you could keep doing that as long as you've got phosphocreatine, which is why at rest you build up a big pile of phosphocreatine that you can use to keep your ATP going when the muscle gets started. But it'll only last as long as you have phosphocreatine. the idea is that the phosphocreatine will keep our ATP from running out until the glucose metabolism starts giving us lots more ATP. So hopefully that makes some sense. Another analogy people sometimes use for this is the idea that if you've got a checking account and a savings account, when you get more money in your checking account than you need, you transfer some of it to your savings account and build up that balance in your savings. Then later, when you're having to spend money quickly and you're draining your checking account, you never want it to go to zero. That's like the amount of ATP you have. So you can quickly transfer money in from your savings account to rebuild the balance in your checking account, keeping your ATP reserves high, while you are still having to spend all that money until you can, for example, get a new job. But you can only do that as long as you have stuff in the savings account. That's like the phosphocreatine. So hopefully that made a little bit of sense. That's the short-term, rapid way of regenerating ATP. Now let's talk about the longer-term methods, that being glucose metabolism. Okay. Now we've talked about anaerobic and aerobic, or glycolytic and oxidative glucose metabolism. So anaerobic, sometimes people call glycolytic, and aerobic, sometimes people call oxidative. I don't like the term glycolytic because really both forms of glucose metabolism start with this process of glycolysis. But let's lay out how this goes. So, our first stage of this, we start with one molecule of glucose, which has 6 carbons. And we're going to start that glucose through a series of reactions where we're going to end up splitting it into 2 molecules of pyruvate, each of which is a 3-carbon molecule. So there's our 6 carbons. It starts out as a 6-carbon glucose and it ends up as 2 3-carbon pyruvates. In the process of doing that, we have to put 2 ATP into it to get it started. But we get out of it 4 ATP. So a net gain of 2. In many cases, people will skip the whole 2 goes in, 4 goes out, and just talk about it produces 2 overall. The other thing that happens here is that 2 molecules of NAD plus are used, and we end up making 2 molecules of NADH. Now, we should take a moment to talk about NAD plus and NADH. So NAD plus you can almost think of as a molecule that has a couple of places on it where we can put things. And what we end up putting on it is effectively what amounts to a molecule of hydrogen and sort of an extra electron. This is skipping over some of the detail. So there's our electron. You could think of it almost as just carrying 2 electrons, one extra and one on the hydrogen. This is now NADH. So NAD plus is the empty version. NADH is when it's loaded. Though that hydrogen and the extra electron those are actually what are going to carry a lot of the energy that we're extracting from glucose in this process. So we did get enough energy to make a net gain of 2 ATP out of that glucose, but we also extracted what amounts to 4 high energy electrons. 2 of them free and 2 each on a hydrogen. I'm just going to talk about the electrons. Don't worry about the hydrogen. So we extracted some energy in the form of those 4 high energy electrons that are being carried on the NADH. The NADH can drop those electrons on other molecules and turn back into NAD plus sometimes. We'll get to that. But This process that we just did right here has a name, and actually, let me see if I can do this on a separate layer. So this particular process, glucose to pyruvate, this we call glycolysis. Okay, so that part is glycolysis. Now, then what? So the pyruvate, we actually have a couple of choices for what to do with the pyruvate. One of them is going to involve the mitochondria. But let's say we don't have enough mitochondria around. So one question that sometimes comes up is, why can't we just keep doing this? What's wrong with making pyruvate? And the answer is nothing. But the cell doesn't have an unlimited amount of NAD+. And every time we take a glucose and turn it into pyruvate, we do get 2 ATP out of it. But we also have now taken 2 of our NAD pluses and made them into NADH. If we keep doing that, we're going to use up all of our NAD+. We'll end up with a big pile of NADH and nothing to do with it. And then we can't do the process anymore because we don't have any more NAD+ to use. So the NADH has to put those electrons somewhere. One option, so I'm going to do this way. One option for what it can do with it is to take those NADH and drop them onto, take those electrons and drop them onto the pyruvate. That will allow us to become NAD+ again. But taking those electrons turns the pyruvate into 2 molecules of lactate, also known as lactic acid. And that lactate is kind of a dead end. A muscle cell can't do very much with lactate. Now, there's a common belief that the lactate or lactic acid buildup is what makes your muscles burn after exercise. And as it turns out, that is actually not true. What does make them burn is probably inflammation and the sort of micro damage you do to muscles when you use them a lot. But the lactate itself is not harmful. It's just the muscle can't do much with it, so it has to be sent off to the liver, which has the right enzymes to use it to turn it, say, back into glucose, to kind of reverse this process. But this is one option. This is one thing we can do with this. And if we end up doing that, there's a name for that one too. That process, going from pyruvate to lactate, is known as lactic acid fermentation. So that is one possible thing that we can do with glucose. We can take a glucose, run it through glycolysis to turn it into 2 molecules of pyruvate, in the process making a net gain of 2 ATP and taking 2 NAD+ and making them into NADH. And then take the NADH, drop the electrons onto the pyruvate so it becomes NAD+ again, and turning the pyruvate into lactate. And then that's the end of the story. That process, glycolysis plus fermentation, is anaerobic metabolism. So if we're going to do anaerobic metabolism, we're going to take glucose, go through glycolysis to pyruvate, and then turn the pyruvate into lactate. The net gain of glycolysis is 2 molecules of ATP from that original glucose, which is not much, but it's something. And it has the advantage that it requires only stuff that's in the regular cytoplasm of the cell, and it's very fast, and requires no oxygen. Notice, no oxygen is necessary in there. So that's the possibility of anaerobic metabolism. But let's talk about the other path that this can take. So what if we have mitochondria? So here, let's draw the edge of a mitochondrion here. So we can bring that pyruvate into the mitochondria and in the process turn it into 2 molecules of acetyl-CoA. Now, to do that, to turn pyruvate into acetyl-CoA, we're going to have to use 2 more NAD+, becoming NADH, and we're going to extract 2 molecules of CO2 as waste products. Acetyl-CoA is a 2-carbon molecule. So now we've gone from 2 3-carbon pyruvates, 6 carbons total, to 2 2-carbon acetyl-CoAs, 4 carbons total. The CO2 are where the other 2 carbons went. That's a waste product there. So this particular step, this one we call, actually, hold on a second. This one we call, it actually has several names. I like the one that we call the linking step. So that one involves pyruvate becoming acetyl-CoA inside the mitochondria. So this is, oops. Nope, wrong one. Wrong one. Stop it. Okay. That should be mitochondrion, singular. All right, so I've moved us over so that you can see a little more easily what's going on. All the stuff we were just talking about, glycolysis and the linking step, are all shown here. Here's the acetyl-CoA we just made. What I've done is move this over so you can see more of what's going on inside the mitochondrion. So a mitochondrion has 2 membranes, an outer membrane and an inner membrane. The space between them is known as the intermembrane space, and the space inside the inner membrane is known as the mitochondrial matrix. Now, we'll get to what these things are over here are in just a moment. What I want to go with is what happens to the acetyl-CoA. Acetyl-CoA runs through this thing we call the Krebs cycle, which is a complex series of reactions that happens in the mitochondrial matrix, where each acetyl-CoA reacts with the end product of the previous Krebs cycle to form to go into the next cycle. We usually draw that something like this. So where the acetyl-CoA comes in, and after a series of reactions, it produces something that combines with another acetyl-CoA to go through the cycle again. Each acetyl-CoA goes through its own Krebs cycle, but since 2 of them come in, we usually draw the cycle and talk about what it produces from those 2, since those 2 came from the original 1 molecule of glucose. The products of the Krebs cycle are 6 NAD+ get turned into 6 NADH. 2 molecules of something called FAD, which is similar to NAD+, become 2 what are called FADH2. 2 ATP come out of it directly. And as waste products, we produce 4 CO2. So each acetyl-CoA goes through its own Krebs cycle. So let's make a note of this. From 1 acetyl-CoA, we get 3 NAD+ and 3 NADH, 1 FADH2, 1 ATP, and 2 CO2. So when 2 acetyl-CoA go in, like they do here, you get double that, 6 NADH and so on. Okay, And notice our 2 acetyl-CoAs, each of which was a 2-carbon molecule. That's 4 carbons. At the end of the Krebs cycle, we've got nothing left. There's our 4 carbons and our 4 CO2. So this finishes the final breakdown of glucose. Glucose has now been completely broken down into smaller molecules. The glucose is gone. So what do we get out of that glucose? Well, my 2 NADH from out here, let's bring them in. So here's 2 NADH from here. I got 2 from the linking step, 6 from the Krebs cycle. So all of these together give me 10 NADH from that original glucose. I've got 2 FADH2s. I've got 4 ATP total. And I ended up with 6 CO2s of waste product. Okay, well, fine and good, but that's still only 4 ATP. And I still need to do something with all that NADH and FADH2. Well, that brings us to the final stage of this aerobic glucose metabolism. So we've done glycolysis, the linking step, and the Krebs cycle. And the end product of all that is 10 NADH, 2 FADH2s, 4 ATP, and 6 CO2. But those NADHs and FADH2s have to put their electrons somewhere to get turned back into NAD plus and FAD. That is going to happen at the last step, which we call oxidative phosphorylation. I'm going to have to redraw this. This is where these things over here are going to come into it. So let me redraw this one more time to focus in on that. So we focused in one more time, and now just on that one spot in the mitochondria where we had those weird proteins. This little set of 4 down here, collectively we call the electron transport chain. I'm going to erase that in a moment, so just be ready for that. Up here, this is ATP synthase. All right, so let's follow this on. I'm going to erase that because I'm going to need that space for something else in a moment here. Okay, so from the previous steps, we had a total of 10 NADH here. They needed somewhere to put their electrons, those high energy electrons they're carrying. That turns out to be the first protein in the electron transport chain. So they come away having turned back into NAD+, having dropped their high energy electrons onto this protein. That protein hands those electrons off to the next protein in the line. And in the process of doing that, it's able to transport some protons, H+, into this intermembrane space. This protein hands its electrons off to here. That one hands its electrons to here. And as those electrons pass down this chain, these proteins can use the energy in those electrons to pump protons into this intermembrane space. So we end up getting a high concentration of H+ in here. Now, what good does that do us? Well, we now have this last protein here, ATP synthase. I should also mention that the FADH2 can also drop its electrons here. If I remember, there's some evidence that it drops them on the second one. And those electrons just get passed down the chain the same way. ATP synthase is here to give these protons a way out. They can diffuse from high concentration to low through ATP synthase, which acts as a carrier protein. But it's a very unusual carrier protein. As those protons move through it, it uses that movement to almost literally turn a little wheel inside that brings ADP and phosphate together to make ATP. It's almost like a little water wheel for those protons. As they pass through, it uses that movement to make ATP, to bring those ADPs and phosphates together. So if we follow along what we did with this energy, we extracted energy from glucose in the form of these high-energy electrons carried on NADH and FADH2. We used that energy to pump protons into this intermembrane space. And then by allowing those protons to leave the space through ATP synthase, we use the movement of those protons to put ADP and phosphate together to make ATP. So we've transformed energy at several steps along the way. But in the end, what we got was ATP. Yay! That was our goal. And we turned our NADH and FADH2 back into NAD plus and FAD, which can then go back and participate in more of the glycolysis and linking step and Krebs cycle reactions. So there's one thing that's missing here. If I'm saying that this is all part of aerobic metabolism, that implies that it uses oxygen. Have you noticed that there's something missing? At no point have we used oxygen. Here's where it's going to come in. Oxygen is going to be the answer to the question, what happened to those electrons that reach the end of the chain? Because they can't just stay there. If the NADH puts some electrons on the first protein in the chain, it has to pass them on in order to be able to accept more electrons. If it doesn't, it's just going to get full and now it can't do anything else. So these proteins pass their electrons from one to another, but if they all jam up at the end, the whole chain is going to fill up. Oxygen is going to help us with that. Oxygen is going to accept those electrons and through some stuff we are not going to get into, come away as water. It accepts those electrons and some protons and comes away as water. Don't try to balance the chemical reaction there. We're not going to get into all of the reduction, oxidation, and all that. It's just we take in oxygen here, we produce water. Oxygen is the acceptor for those electrons. It keeps the electron chains from jamming up. The poison cyanide is poisonous because it actually prevents oxygen from getting those electrons. If you get poisoned with cyanide, your cells can't do this last step because all of those electron transport chains jam up because the electrons can't get taken off of them. So you can't do this process, so you can't make enough ATP through aerobic metabolism, and your cells can't make enough ATP to stay alive. Okay, so that was our final step. That's oxidative phosphorylation. We use the energy from the electrons to pump protons into that space. Oxygen then accepts those electrons, makes water. The protons diffuse out through ATP synthase, which uses that movement to make ATP. How much ATP? That's actually going to depend on how efficient this is. Unlike the other steps, where I could say glycolysis produces a net gain of 2 ATP, always 2. And the Krebs cycle produces 1 ATP for every acetyl-CoA that goes through it. Here, the number produced is actually not constant because there's too many steps where things can happen. For example, some of these protons can get out of this intermembrane space through other means. So exactly how many ATP I get, that's a little variable. But as a rough estimate, one NADH provides enough energy for about 2.5 ATP most of the time. Overall, when we account for some of the losses in this process. One FADH2 isn't quite as efficient, and we only get enough energy for about 1.5 ATP. So let's see how many we got from this. 10 NADHs, which is what we came into this with, times 2.5 each, means that for each original glucose, I'm getting about 25 ATP just from the NADH that was holding those electrons. 2 FADH2 times 1.5 each is about 3 ATPs worth. So this step, oxidative phosphorylation, gives me enough energy to make about 28 ATP for every glucose that started this whole process. All right, now let's put all of this together and make a summary chart of all of the steps, what goes in, what goes out, and what we get from them. So here's our chart. For each step, we're going to look at what goes into it, what useful things come out, that's the output, what not useful things come out, that's the waste, things we can't use to get more energy. And then how much NADH we make or use up, how much FADH2 we make or use up, and how much ATP we get. So in our first stage, glycolysis. Actually, our first stage, the input was 1 molecule of glucose. The output was 2 molecules of pyruvate. There were no direct waste products produced in this step. So nothing there. I made 2 NADH. I made no FADH2s. And I had a net gain of 2 ATP. If you wanted, you could have as the input, you could put 2 ATP there and have 4 as your ATP produced, but I'm just going to keep it with the net gain right now. Right now, let's do the aerobic pathway, not the lactic acid fermentation. Let's assume we have mitochondria and we're going to go into that linking step. So the next stage was linking step. What went into that was the 2-pyruvate. The useful things that came out from the linking step were 2-acetyl-CoA. If you remember, in the linking step, we made waste of 2 molecules of CO2. We also made 2 more NADH, no FADH2, and there were no ATP produced directly in the linking step. Okay, next was the Krebs cycle. What went into that were the 2-acetyl-CoA. And in terms of useful output, there wasn't any. This was the final breakdown. So we ended up making 4 CO2s as waste products, but no useful output from that one. We did, on the other hand, make 6 NADH, 2 FADH2, and 2 more ATP. Then we had oxidative phosphorylation. Now, we didn't do the balanced chemistry here, but I'll tell you that in order to take those electrons off the chain, we are going to need 6 molecules of oxygen. Those are the final electron acceptors. There's no particular useful output, but for waste, we're going to get 6 water from those oxygens. We're going to put all 10 of those NADH in there. So I'm going to represent that as using up 10 NADH, which means we get 10 NAD plus back. Likewise, we're going to use up the 2 FADH2s and get FAD back. And if you remember, we got 25 ATP from the NADH and 3 from the FADH2, so that's a net gain of 28 ATP. So what's our end product here? The pyruvate we've accounted for when it output from one and input to the other, so it kind of cancels out. The acetyl-CoA likewise. So our total input for aerobic metabolism was a glucose. That should be a G, not a 6. And 6 oxygen. The waste produced was 6 carbon dioxide and 6 water. Total NADH balanced out in the end, as did FADH2. And in the end, we made about 32 ATP. So the total equation in the end of this for aerobic metabolism is glucose plus 6 oxygen produces 6 CO2 plus 6 water. That actually balances out. The equation for glucose is C6H12O6. That's the formula. So you can actually see that this equation balances as if you go track all the carbons, oxygens, and hydrogens. And in the end, about 32 ATP worth of energy. That's what aerobic metabolism of glucose does for us. What you need to be able to talk to me about what happens at each step, how many NADH, how many FADH2s are produced, how much CO2, where the ATP comes from, what's necessary at each step. You should be able to talk about all of those. You should also be able to do this for anaerobic metabolism. So actually, let's do that. For anaerobic metabolism, we had an input of a glucose, so this is in glycolysis. Input of a glucose, output of 2-pyruvate. No waste at that point. We produced 2 NADH, no FADH2, and 2 ATP net gain. Then, if we didn't go down the aerobic pathway, we ended up doing lactic acid fermentation. Which took our 2-pyruvate and produced 2-lactate as waste. It consumed the 2 NADH, because remember they gave their electrons to the pyruvate. No FADH2, and no ATP is produced in fermentation. So our net result there for anaerobic metabolism, net input was glucose, really nothing else, produces 2-lactate, and 2 ATP worth of energy. Much less efficient, but much easier. This doesn't need the mitochondria, it doesn't need oxygen. It happens outside in the cytoplasm, it's very quick, it just doesn't produce much energy. Whereas if I switch back to the one for aerobic metabolism, it needs oxygen and it requires the mitochondria, and it's more complicated, it's a slower process, but we end up producing a lot more ATP from it. Looks to me like, 16 times more? So, that's the idea of anaerobic and aerobic glucose metabolism. Now, in the last part of this lecture, we're going to talk about fatty acid metabolism, which is related to aerobic glucose metabolism. So, I'll see you in there.